All right, so first thing I need you guys to do is double check, make sure you have all your answers or your questions answered on your chromatography lab from the leaves, and then make sure that you get that submitted back to me. If you're waiting on your picture, let me know and I can always send it to you. I did pull out the last few ones yesterday from A group that we're still running. So today we're going to expand a little bit more on our vascular tissue and what it means to be a monocot or a dicot. Remember that vascular tissue is going to be the tissues that allow for the transportation of water as well as the transportation of the sugars that are produced in the leaves, and it goes back into the actual tree or plant and then down to those roots. The water goes the opposite way, it goes from the roots all the way up through the plant out to the leaves. The xylem, remember X is really close to W. That's how I always remember this, is that W, water, really close to X in the alphabet. So the xylem is responsible for conducting water through cohesion. So that means that the water particles actually stick to the other water molecules, and that helps it climb up the tree. And then also sticks to the parts of the vascular bundles using adhesion. So hopefully you've heard those two terms before, the cohesion and the adhesion, but it will actually stick to the sides as it moves up and up and up, so it makes it move a little bit quicker. Remember, water is needed in photosynthesis. The phloem, how I remember this, P is really close to S, is the conduction of the sugars made in photosynthesis from the leaves all the way down into the tree or plant to the roots for some storage. This happens again as a form of food storage. During the winter, remember those leaves are going to fall off, so any sugars that were produced in the leaves have to be out of the leaves. We can't store any sugars in the leaves, so they try to gain any nutrients, squeeze out the last little bit that they possibly could from that plant. If I take a look at a vascular bundle, that vascular bundle includes the xylem and the phloem. You might notice that the circles here, coming through the circle, would be the water. Okay, so it is an opening. Picture it as if there's a bunch of straws through the middle of the plant, and in those straws you're conducting water and sugar. So the conduction of water is a little bit different than that conduction of the sugars. Okay, the sugars actually are a lot smaller and a lot lesser in quantity. So if you look, the openings for the phloems are a lot smaller than the openings for the xylem. We are going to be looking at this under the microscope on Friday for B group and Monday for A group. So when we look at most angiosperms are going to be our flowering plants. They get divided into two main groups, and those are the monocots and the dicots. This is important in terms of taxonomy. Remember all the way back from week 1 when we talked about how we classify every living organism. Remember, viruses are not on that same category. They are not in the same taxonomic levels. They're a little bit different. But when we look at the early zygote, which is going to be that seed as it germinates, it's going to germinate from that embryo and produce its very first root and its very first leaf. Those cotyledons are the very initial seed leaves. So monocots have one cotyledon that is initially produced. Dicots have two cotyledons that are initially produced. And there's a couple examples there for you. So again, if you take a look as it's growing, you're going to see, are there two leaves that are popping out as it germinates, or is there only one that is popping out? In addition, one quick easy way to identify whether or not a plant that has already germinated, that has many, many leaves, is a monocot or dicot, is by looking at its venation patterns. Monocots tend to have parallel venation, and dicots tend to have a netted venation. Additionally, we can look at the roots and say, okay, well, what type of roots do they have? Monocots are usually very, very fibrous, and they kind of extend out. They don't go very deep into the surface. So you think about grass. When you rip grass out, you see all those short little roots that kind of go side to side throughout the top portion of the soil, whereas dicots have a taproot that extends all the way down. Additionally, on that taproot, you can see smaller roots going out to the side, and then it also has what looks kind of like a hairy, those are root hairs, those actually increase the surface area of the roots that allow for them to absorb more water. For our flowering parts, if we look at the actual petals or we look at the reproductive structures, monocots always come in groups of 3s or multiples of 3s. So if we see 3, 6, or 9, it is typically going to be a monocot. If for a dicot, typically comes in groups of 4s or 5s. Everybody always asks me, well, what about 15? What if there's 15 petals? Well, would it be a dicot or monocot? Rather than just counting the petals, we could go and take a look at other parts of the plant and say, well, is it parallel venation? Is it fibrous roots? All these other characteristics. Or we could also look at it under the microscope. So again, the vascular bundles, this is what we're going to be looking at in the lab. For monocots, if you look, those vascular bundles are scattered throughout the entire plant stem. Right? Whereas for dicots, it is just along a one circle, okay? So around that side. So as we look at that under the microscope on Friday and on Monday, you're going to be able to see sort of what is that structure, how big are they, what happens with the vascular bundles, is there more phloems, is there more xylem, why are they a little bit thicker for the water, the xylem, versus the phloem. Stem types, if you take a look, most monocots are going to be herbaceous, so that means they're going to be pretty much green, okay? Whereas dicots can either be green or they can also have woody tissue. Here's a better view of the seeds. So if you look, the 2 here are the cotyledons. So those seeds, if you take a look and you cut them in half, you know a bean has 2 parts of its seed. Whereas if you look at a corn kernels only have 1 portion, okay? So even if you cut it in half, you wouldn't see 2 distinct parts, unlike like a lima bean. Here's a better view of what a lima bean looks like. Those lima beans, again, can kind of flake off into their 2 parts, their 2 cotyledons. They do have an external protective coat, that is the seed coat, that you can see at the bottom right picture. That just kind of helps protect it as it germinates so that then it doesn't get damaged by other plants or animals, and then it can actually germinate. You can see that early embryo growing right there at the bottom as well. All right, so for just a quick wrap up again, monocots, dicots, again, this is part of our flowering plants. Our monocots, again, coming in pairs of 3s, so 3, 6, 9. For our flowering plants that are dicots, they come in 4s or 5s. Vascular bundles, again, scattered throughout for a monocot, and going to be in 1 distinct circle for our dicots. Monocots having those parallel leaves, and dicots having their net venation. Here again is a chart for you that you can use to help you in the next couple of pages. So on these next ones, I want you to take a look at them and try to guess, are these monocots or dicots, okay? All right, so take a look at this one. If you look, there are 2, 4, 6, 8 flowering parts. So you should note that that is a multiple of 4. That would be a dicot. Take a look here at this fruit. There are 5 chambers of seeds, 1, 2, 3, 4, 5. So this is a dicot.
What's your new last name?
All right, so take a look at this picture. You have 3 petals, but the male reproductive parts, the anthers, you can see they have pollen on them, comes in a 6, which is a multiple of 3. This one is a monocot. Here I have parallel venation, and that would be a monocot. Vascular bundle arranged in a circle, that would be a dicot. Take a look at your petals, 1, 2, 3, 4, 5, 6. It's a multiple of 3, it would be a monocot. Look at our tomato, there's 1, 2, 3, 4, 5 chambers. Tomatoes are dicots. We have a net venation. This would be a dicot. Vascular bundles spread throughout the entire stem, monocot. And then again, this is just an extra sheet for you to fill out that you can use as notes and hang on to for future assignments. And then when you guys come on Monday, we're going to be doing a carnation activity where you can actually see and change the color of carnations and look at the transportation of water up and how it goes out to the petals. And then we'll also be doing a lab on Friday, Monday as well. So those days you're going to be looking at monocots and dicots under the microscope. Hope you guys have a good rest of your day.